So on the paper I have a hypothetical developmental network for the formation of spikes on certain cells of the ectoderm of a spiny beetle. As you can see, S it activates P, and you can tell that it activates it because of the arrow, and then P activates I. Now I represses K, and you can tell that it represses K because of the bar. K represses E, which then creates spikes. E creates spikes. The reason why spikes are formed is because since I represses K, K cannot repress E. However, if there was to be a mutation in P, this means I would be unable to form, which means K is allowed to inhibit E, which means there'd be no formation of spikes. If we had a mutation in I, that means I cannot inhibit or repress K from forming, which means K will repress E from forming, which will repress spikes. If both I and E are repressed, it will not create spikes for the same reason that a mutant I strain will be unable to produce spikes because I will not be able to repress K, which will then repress E, but also because E is not able to create the spikes. So if these two are mutants, spikes would be unable to be made. However, if we were to have a mutant S and a mutant K, then there would be no S to activate P, which will then activate I, which will then inhibit K. However, there is no K as well to repress the formation of E, which means that E is actually able to form spikes, unlike our other three up here. And one hypothesis as to why not all cells in the ectoderm have spikes is due to cell differentiation. And K probably produces another type of cell that is just as important to the organism, but also cannot function if it produces spikes. So K inhibits E to inhibit the production of spikes, but then activates the production of something else that is important.